Okay, so welcome to this video in which we are going to begin our discussion of endothelium-dependent hyperpolarization, and this video is going to be in the cardiovascular system playlist. Okay, so we are going to start our discussion of the endothelium-dependent hyperpolarization, which is something that we're going to now do a series of videos on and look at all the different processes involved in it. And it's fair to say that this is a complicated pathway. There are absolutely loads of intertwining pathways that all seem to be playing a role in this. In this video, we're just going to introduce what it is and see a few experimental correlates that first showed its existence. So endothelium-dependent hyperpolarization. And endothelium-dependent hyperpolarization, which is slightly squashed over into that side, is also often referred to as EDH, endothelium-dependent hyperpolarization. Okay, right. So, what it is then? So, it was first discovered by a group led by a scientist known as Bolton. So this is Bolton et al. And this was in 1984 that it was first seen as a phenomenon, basically. Bolton et al., 1984, were the first people to actually find endothelium-dependent hyperpolarization. So if you're not used to seeing references, et al. Means Bolton and colleagues, basically. Right, so what was he doing? Well, basically, what he did is he took mesenteric arteries from rats. Okay, so the mesenteric artery is just a blood vessel which supplies the intestine. And it's a nice blood vessel to work with because it's a high-resistance blood vessel. The mesenteric bed is a very high-resistance bed of arteries. So they're quite small little arteries. So we'll have a look at what he did. Okay, so what he got was he got one of these arteries from the mesenteric bed here. Okay, so let's discuss the structure of an artery, or of any blood vessel. It's an arteriole, really, that we're talking about here. A little artery. Okay, so we have endothelial cells lining the lumen of the blood vessel. So here are our endothelial cells, and they sit on the basement membrane. Okay, so the basement membrane is a sheet of collagen, basically, that these endothelial cells are sitting on. And this is the sort of longitudinal view. Let's also draw down here the cross-section view. So here's the tube that the blood is flowing through, and basically these endothelial cells are sitting on this tube, like so. Well, they're sitting on the basement membrane, lining the lumen of the tube. Okay, so these are all endothelial cells. So we have the nuclei of the endothelial cells here. Now, let's draw the basement membrane which these endothelial cells are sitting on. Let's have the basement membrane in this turquoise colour here. So this turquoise sort of surround for the endothelial cells, this represents the basement membrane which the endothelial cells are sitting on. So I'll draw this in this picture as well. So here is the basement membrane that the endothelial cells are sitting on. Okay, and then surrounding the basement membrane, what you then have is the subendothelial space, which is a space full of connective tissue. And then after that, what you have is the internal elastic lamina, which is made up of elastin, basically, which is a very elastic fibrous protein. Okay, so let's draw the internal elastic lamina in blue, which is surrounding this subendothelial space here. So this is the internal elastic, oops, missed it there. There we go. Right, so let's label bits up then. So in turquoise here we have the basement membrane, okay, which is a membrane of collagen. So this is the basement membrane, also shown over here, okay. Then surrounding it, this space which I've left empty, this is the subendothelial space, or the subendothelial connective tissue. And again, it's mainly collagen that you have in the subendothelial space. Okay, and then surrounding the subendothelial space, you then have here the internal elastic lamina. Okay, which is a sheath made up of mainly elastin fibers. Internal elastic lamina. Right, now let's add the internal elastic lamina into this longitudinal picture here. So we have the subendothelial space after the basement membrane, and then you have the internal elastic lamina outside of that. So in blue here, this is the internal elastic lamina. Okay, right. And then surrounding that, what you have is rings of smooth muscle cells. Okay, so let me show these. Here are these rings of smooth muscle cells, like so. Okay, which surround, they encirculate the blood vessel within. Okay, and it's going to get a little bit difficult over here. Okay, so they are oriented circularly around the lumen of the blood vessel in these sort of rings. Right, now this makes them quite difficult to show on this longitudinal picture, because when you look at them in longitudinal, all you're going to see is the little cross sections of them, because what you've effectively done is cut down there, and you're just going to see the belly of these smooth muscle cells. So maybe this is a ring of smooth muscle cells, and then you might have some more here, so they're going to look like this basically. Okay, so this is this smooth muscle layer. Okay, and the smooth muscle layer has a name. It's known as tunica media. Okay, and it's bounded on the outside by another elastic lamina, which I'll show here, or here on this drawing. Okay, so let me put in this other elastic lamina, and this is the external elastic lamina. Okay, so let's put this in red here. So in red, this is the external elastic lamina surrounding the layer of smooth muscle cells. And basically, the layer of smooth muscle cells up to the external elastic lamina, this is known as tunica media. Tunica means layer, media means middle, so it's the middle layer. The endothelial cells all the way up to the internal elastic lamina, that is the tunica intima. Tunica again meaning layer, intima meaning close to the blood, so the layer that's close to the blood. Okay, so let me colour in the external elastic lamina in red here as well, and then we'll label it as well. So here's the external elastic lamina. And then external to the external elastic lamina, you have a layer of connective tissue known as the tunica adventitia. Okay, so this here, this is the external elastic lamina. Now, we have missed off this tunica adventitia, but it's not going to be terribly important for our discussion of endothelium-dependent hyperpolarization, so I don't feel too guilty. External elastic lamina. So the final layer that you just have to put in to complete it is the tunica adventitia, which is connective tissue. Right, okay, so endothelium-dependent hyperpolarization. What Bolton et al. Were doing was that they were stimulating these endothelial cells here with either acetylcholine, or alternatively you can use carbachol, which is a non-selective agonist for acetylcholine receptors. Okay, so you can use carbachol or acetylcholine. We'll say we'll use acetylcholine. So acetylcholine will bind to the M3 receptors, which are on these endothelial cells here. So this is the M3 muscarinic acetylcholine receptor. So M3 equals a muscarinic, little m, acetylcholine ACH receptor R. So basically what they were doing was they were stimulating with acetylcholine, and what did they notice? They noticed that if you record the voltage across the across these muscle cells here, if you ask what is the electrical potential difference between the extracellular and intracellular compartments, let's draw one of these smooth muscle cells larger here. So basically what Bolton et al. Were doing were they were measuring the electrical potential difference. Let's put a voltmeter here. They had an electrode stuck in the extracellular fluid, and they had an electrode stuck in the intracellular fluid. And what they were asking was, what is the electrical potential difference from extracellular to intracellular? Which means, what is, if you had a little man standing in the extracellular fluid, and he measured electrical potential in the extracellular fluid, so let's call that the electrical potential of the extracellular fluid, and he was to move into the intracellular compartment and measure electrical potential there, let's call that the electrical potential of the intracellular fluid. Basically, this voltage or electrical potential difference from extracellular to intracellular means if you were to move from extracellular to intracellular, what change would you see on the machine measuring electrical potential? Okay, so that's the electrical potential difference. So basically it's the new electrical potential, the electrical potential inside, minus the electrical potential extracellular. Okay, and this is usually, well, it's usually in muscle cells around negative 85 millivolts, okay? So basically, what they saw was a hyperpolarization of this smooth muscle cell in response to the acetylcholine. So they saw the electrical potential difference across this membrane get even more negative, i.e. the amount by which the intracellular electrical potential is lower than the extracellular electrical potential got even greater. So they saw a hyperpolarization in response to the stimulation of the endothelial cells with acetylcholine. Okay, right, we'll continue this discussion in the next video.